This year, we're delighted to celebrate the 87th Paget Lecture, adding Professor Robin Lovell-Badge to our long and eminent list of lecturers. Robin is a principal group leader and head of the Laboratory of Stem Cell Biology and Developmental Genetics here at the Francis Crick Institute. Robin's research in the fields of embryology, stem cell biology, and mouse genetics has contributed to groundbreaking discoveries. These began with contributions to the development of embryonic stem cells from mouse embryos and to transgenic mice, both of which proved ways to derive genetically altered animals that were widely adopted. He then went on to discover SRY, the Y chromosome gene responsible for initiating testicular development in mammals, and to characterize how it functions. At the same time, his group found the SOX gene family, including SOX2, which they showed was important for pluripotent cell types in the early embryo, for embryonic stem cells, and for embryonic progenitors and stem cells in several systems in both embryos and adults. Because of his research, Robin has a long history of talking to the media about animals and research. He's also very active in both public engagement and policy work around stem cells, genetics, human embryo, and animal research, and in the ways science is regulated and disseminated. He's always been a strong advocate for openness in the Concordat and is a former council member of Understanding Animal Research. In 2018, Robin was awarded a CBE for outstanding contributions to science. Tonight, I'm delighted to introduce Professor Robin Lovell-Badge and his lecture, Navigating the Complexities of Working with and Talking About Animals in Research. Thank you, Robin.
Well, of course, I'm greatly honored to be giving this talk. I feel obviously very passionate about the whole openness issue, as I'll mention towards the end of my talk, not just about animals and research, but more generally. And but just to say, thinking about all these issues already, it brings back so many memories, the bad and the good. The bad being, when I was working at what was the MRC National Institute of Medical Research in Mill Hill, the weekly demonstrations we used to have outside. I've been, to my sins, president of the Institute of Animal Technology for 19 years, I think it is now, coming on to 20, far too long. I remember when I first used to go to the annual meetings that there was always a big police presence and worry that there was going to be quite violent demonstrations and things. After a few years, basically, that all went, and it was the Concordat that had a big role in that. I'm very honoured to say that I was actually both on the on advisory group for Science Media Centre and on the Council of UAR when the Concordat was developed. So I feel I made a small contribution to that, for which I'm very proud. Right, so I'm going to tell you a little bit about my research and a little bit about other things, sort of extra curricular activities, so you get a flavour of the sorts of animal research we do, as well as the other things I've been interested in. And let me see if I get right way. Yes. I just wanted to point out, this is on the website. First of all, I have a beard, and I don't know why the photo wasn't used me with a beard. But also, that I am not the mysterious animal behind the COVID-19 pandemic, which I on the first this page.
You chopped off the title.
Okay, and of course last year's Paget lecturer talked about sex as well, so you're going to get a bit of sex this time, so maybe it's a common theme. And you've got reproductive organs on your awards, so it's a major thing that UAR thinks about. Okay. So just a little bit of my history. I mean, I'm going to talk mostly about the sex determination work that we've done. But just to point out that, as was briefly mentioned in the introduction to me, I've also done other things. At the same time, we cloned the SRY, the gene on the Y chromosome that triggers male development. We also found the SOX gene family, and that included SOX. So I should say I worked as a PhD student, as a postdoc with Martin Evans, working initially on embryonic carcinoma stem cells, and then towards the end then on embryonic stem cells, which Martin derived in the lab. These became a very important route to making genetically altered animals. And I should just point out, where are we? Martin Evans got a Nobel Prize for this. Very proud he got a Nobel Prize for it. He got that in collaboration with others for gene targeting, so it was an important route to that. So we cloned, when we cloned SRY, this was 1990, we also found this other family of genes that included SOX2. We showed that SOX2 was involved in early development in the mouse, it's critical for pluripotent cells, including embryonic stem cells, which is interesting. I sort of backtracked to what I started with. Shinya Yamanaka won a Nobel Prize for showing you could use SOX2 and other genes to reprogram somatic cells to give induced pluripotent stem cells, which of course is another very important development. I've missed out, but I never expected it, so it's all fine. Both Martin and Shinya deserve their prizes. We've gone on over the years to look at other aspects of stem cell biology, and not just in the early embryo. We looked at the role of these genes, particularly the SOX gene family, in central nervous system development in neural stem cells, where SOX2 and another related gene, SOX9, are very important. Also in pituitary development and stem cells in the pituitary system, and that's occupying quite a lot of our time at the moment. So we're still actually doing a lot of research on this. So back to the main topic. So just a couple of definitions. So when I refer to sex determination, I mean the decision to develop as male or female and how that switch operates. Then you can talk about sex differentiation or gonadal differentiation, which is how that decision is translated initially into making ovaries or testes, and then all the differences between the sexes, or most of them at least. So why do research on this? So pure interest. Of course, there have been theories of how you have males and females been around for actually thousands of years. So one of the ancient Greek philosophers, by the name of Anaxagoras, proposed that semen from the left testis gave rise to girls and from the right testis to boys. That's a theory that can be tested quite easily. That's not true. It's obviously socially important, and often in the news these days, differences between males and females. So I'm not going to go into all of that. So it's a paradigm of how decisions of cell fate are taken during embryo development. Often cells during development have to choose whether they're going to go in this type of cell or this type of cell. For the gonad, you've got these bipotential cell types, so it's a beautiful system in which to study that. It's also a paradigm of how you make organs. The gonads are relatively simple organs, and so it's a very good topic to look at. Clinically important, because there are in fact many cases where the whole process goes wrong, generally referred to as disorders of sex differentiation. That can be complete sex reversal, where the individual develops the wrong sex compared with their chromosomal makeup, for example, XX males or XY females. You can have where you have a mixture, so intersex cases. Inevitably, these lead to infertility. So these are of great concern, obviously, to the individuals who have them and their families and the doctors who have to look after them. They're very difficult to deal with. And while many of them are rare, when you sum up all the different causes, it's quite a lot. It's something like 2% of births. So it's really a lot of cases of DSDs. Some of them relatively mild, but many not. I'm not going to talk about issues about gender dysphoria, although it may be relevant. There's obviously many conditions, including cancer, with differences between males and females. And I guess last year's talk was a lot about that, I believe. So I'm afraid I missed it. So knowledge of the genes and processes involved allows better diagnosis, counselling, appropriate choices of treatment, and perhaps development of novel clinical options. And that's what we always have an eye out on that. So a little biology lesson. Many different mechanisms have evolved as the switch to trigger male versus female development. And we can divide these broadly into two classes. Those environmental switches, where all individuals within a species have the same genetic makeup generally, but it's the environment which influences which sex develops. And you see this in many species of fish and reptiles. And reptiles, many of you probably know, it's temperature at which the egg is incubated, the embryo is incubated at a critical point in its development that turns between male and female. I'm not going into that. My ex-postdoc of mine, still a colleague working in the US, has worked out the mechanism for turtles. And it's beautiful science. So if you're interested, you should look that up. Blanche Capel. Okay, so phenotypic differences between the sexes must depend on either the environmental trigger or hormones made by the ovaries or testes. It's very clear. And then you've got chromosomal or genetic-based mechanisms where males and females differ in one or more genes or in whole chromosomes or sets of chromosomes. So I'm not going to go into talk about birds, but birds, the females are ZW, the males are ZZ. Mammals generally are XY male and XX female. So it follows that in almost all species we know that the Y chromosome acts as a dominant male determinant. So it doesn't matter if you have multiple X chromosomes. If you don't have a Y, you're female. If you have a Y chromosome, irrespective of the number of X chromosomes, you develop as a male. There's some, and apart from the ones on your right, I assume they're normal XXXY, male and female. There's something slightly different with each of those animal species illustrated, which is not quite consistent. So marsupials, it's true whether you make testes or ovaries depends on whether you are XX or XY, but whether you make a pouch or a scrotum depends on the number of X chromosomes you have. Moles, the females have a period where they have a high level of testosterone made, and they have part of their gonad looks much more like a testis. I guess you have to be fairly aggressive if you're running around a dark tunnel. You don't know who you're going to meet. Wood lemmings actually they are different. So the three different species of wood lemming that have been studied, and they don't necessarily have they don't have a Y chromosome necessarily. So some of them have lost a Y chromosome. We don't yet know how sex determination works in those. And then hyenas, they have an XXY system, which determines whether they make ovaries or testes. But the females have external genitalia that's very masculine. So I don't know how they would be treated in debates about which toilets you should use these days. They all have phalluses. So, and if the females want to start wincing now, of course they have to not only have sex through the phallus, they have to give birth through this. So it's a complicated story. But I don't recommend being a hyena. And female, at least. So genetic sex is established at fertilization. The inheritance of an X or Y chromosome from the father is not the whole Y chromosome that determines maleness. It's this gene we found on the Y chromosome termed SOY. I'm not going to go into all the history, but it's a little bit of history. It was 1959 that the Y chromosome was shown to be sex determining in mouse and man, same year. There was a theory for a long time which suggested that it was an antigen that occurred on the surface could be detected on the surface of cells from XY individuals, but not XX individuals, called HY. But that was disproved by Anne McLaren, who showed that and Lewis Simpson, who showed that it can't, because they found male mice that didn't have HY. There was a gene proposed to be the sex determining gene on the Y called ZFY, proposed by a lab in the US, led by David Page. Everyone believed that for a couple of years. I didn't. I was very persistent, and with a collaborator, first of all, we started looking at what this gene was doing in mice, and it clearly was not responsible for sex determination for a number of reasons. And then with Peter Goodfellow's lab, we found a gene on the Y that was a very, very good candidate. We call that SRY. That was 1990. in 1991, we did some transgenic mouse experiments that proved that it was the gene, and the only gene from the Y required to trigger testis development and mostly male development. So you this is an XX male mouse carrying copies of a mouse SRY gene as a transgene. His name was Randy. This triggered a lot of interest from the media. So this was really my initiation into talking to journalists and the media. We had basically for weeks and weeks we couldn't do any work in the lab because we had journalists there. It was crazy. So anyway, I got very used to how they work and what the issues are, and so this was 91. And so since then I've not been frightened of talking to the media. Some of the headlines are rather weird, by the way. I should point out, there was only one bad headline here, which you might be able to see, of mice and Mengele. That was in the Evening Standard, and luckily I didn't have to do anything about this. Blanking his name, really bad. Someone else very involved in public engagement with science called up the editor, and the next day they published an apology. So it was really, really bad. Lewis Walter, sorry, I should remember that. Right, so a little bit about sex determination and gonad differentiation. So if the pointer here was that, it does work, good. So this is in a mouse at about 11 and a half days, where you can't see any visible difference between male and female gonads. In humans, that would be around about 6 weeks of development. And so if you are XY, or particularly you have an SRY gene, that leads to the formation of testis, and you can see distinct differences already just 1 day later between the ovary and the testis here. There's this prominent blood vessel, but the most important part is that you have this sort of stripy appearance, which is due to differentiation and alignment of supporting cells, precursors, into Sertoli cells. So Sertoli cells are characteristic of the testis. So these organize around you have the germ cells, you have a layer of Sertoli cells around these, can't point very easily, very thin layer of pituitary myeloid cells, which help make this sort of testis cord structure, which is again going to go on to form the seminiferous tubules much later. And then in the interstitium you have a variety of cell types, but particularly you have the Leydig cells, which make testosterone. In the ovary, not a lot is happening in terms of organization. You basically have germ cells surrounded by pregranulosa cells, roughly at random at the beginning. But we actually know that molecular changes are already initiated on the pathway to making an ovary. So SRY is active for actually a very brief period. It looks sort of extended here, and it peaks at around 11 and a half days and then goes off. So the first time you start to see overt differences, SRY is no longer active. And if you look on a cell-by-cell basis, SRY is only active for about 3 or 4 hours. It's only present for about 3 or 4 hours. So it acts very rapidly. And its role is basically to flick a switch. That's all it's doing. The whole process is a balance on a very fine knife edge, and it just requires a little bit of SRY activity to trigger things to go the testicular pathway. And if SRY is not there, it follows the ovarian pathway. So SRY diverts the ovary pathway to make a testis. Once you start making a testis, this produces a number of factors like testosterone that then masculinize the rest of the embryo. So this is that decision basically. You have supporting cell precursors, which will in the ovary give granulosa cells, in the testis give Sertoli cells. And then once they start making Sertoli cells, they influence all the other cell lineages in the gonad to follow the testicular pathway. That includes the germ cells, steroidogenic cells, etc. So that's the initial decision is taken there. So what does SRY do? Well, all the evidence is suggesting that it's the only critical gene it regulates is a related gene called SOX9. The SOX9 is actually involved in many systems during development and in the adult, and I mentioned it's expressed in a number of stem cells and other cell types in the adult. But it's particularly important in sex determination because it's a critical gene for making Sertoli cells. So we know this from human mutations and from mutations in the mouse. So loss of function mutations can give you XY females. We also know that if you deliberately misexpress SOX9 during mouse gonad development, then you can get XX male development, so no SOI there, but you get male development. And there are rare duplications of SOX9, or as I mentioned in a minute, a specific enhancer region you could also find in XX males. So you just need a little bit more SOX9, and that's sufficient to trigger male development. Right, where are we? So it turns out that many these disorders of sex development sex differentiation are due to altered SOX9 gene regulation. Not all of them, but certainly many of them are. So we spent a while looking at how SOX9 is regulated, and I'm going to cut a long story very, very short and just say that we found one specific enhancer, which we called enhancer 13. It was basically the 13th one we found, which is just 557 base pairs long, located a long way from the transcriptional start site of SOX9. And if we deleted this, 100% as homozygous deletions, 100% of XY animals developed as females instead of males. So we have XY female sex reversal. And this is just showing some of the gory details. So this is a normal male control, if you like, with testes. This is a normal female control with ovaries. And this is our homozygous mutant for the enhancer, where you have structure a bit smaller than normal, but they look much more like ovaries. So this little region of DNA controlling the activity of SOX9 is critical to make testes. So that fits with human data suggesting that deletions of the region where this equivalent to this enhancer is located are associated with XY female sex reversal, and with duplications of the same region that's been found in some XX males. So it's a critical little enhancer. And just unpublished data, but it's Angona who did this work in my lab, has gone on to study this in a lot more detail. And she's shown, in fact, that just altering one or two base pairs within this enhancer can be sufficient to give you sex reversal. It's incredibly amazing that just a tiny bit of DNA, and this is a non-coding region, a tiny little bit of coding region is so specific. So there's the mice. Well, one is a control, one is an XY female. I don't know which is the control, actually, but they both look female. And the reason for this is basically you've reduced the level of SOX9 expression. So again, this is our normal male. This is the level of SOX9 expression. So you have high expression here. This is our if you like, control female, low level of SOX9. And when you mutate the enhancer, you have the same low level of SOX9 as you do in the control female. And the opposite, if you look at markers of ovary development, and FOXL2 is a very critical one I'll talk a bit more about later. That's high now in our XY females. And we can also show you molecularly that SRY is bound to enhancer 13 at the right time and place in the developing gonad. So again, I'm not going to bore you with all these technical details, just to say that we can physically show that the two interact. So SOI interacts with enhancer 13 just to boost the expression of SOX9 enough that it reaches a critical threshold, that it then drives the Sertoli cell differentiation. So this is what I mean. So you have this everything balanced very finely to begin with, and we know from looking at gene expression, gene activity at this, in different gonads, or these early stages of the formation of the gonad, that there is very little difference between an XY and an XX early gonad. In fact, no difference. They're identical. But all you need is this little bit of SOI expression to boost the expression of SOX9. So SOX9 is expressed here, but at a very low level. You boost the expression of SOX9, and then it triggers these sort of feed-forward loops, these pathways that are going to initiate cellular cell differentiation and then maintain it through testis development in the embryo. In the absence of SOI, SOX9, this low level of SOX9 goes off, and it goes off quickly. There's a molecular changes going on in here, and we know of a number of genes that are involved in this active process of making an ovary. WT1 is involved, Wnt signaling pathways, and this gene FOXL2. But they act in a redundant way, so making mutations in any one of these doesn't necessarily give you a dramatic phenotype. FOXL2 is an interesting gene, and we've been devoting a lot of time to looking at this because over the years, my lab and many other labs now have generated a lot of understanding of the genes involved in making a testis. We wanted to push what makes an ovary, and I mentioned a few of the genes involved. And this one, FOXL2, caught our eyes because it was actually it's the beautiful marker of early ovary development. So you can see this is staining for this is a transgene reporter, and you can see its expression very clearly in the XX, not the XY gonad, and it's the earliest marker, clear marker we have of these pregranulosa cells in the early ovary. Now, it was found as a candidate for a gene giving XX male sex reversal in goats. Now, why were people looking at goats, you might ask? So goats have horns. So farmers quite like their goats not to have horns because they damage things, they damage each other sometimes. There's been a couple of mutations that have occurred spontaneously that the farmers have been keen about, and one of them called POLD, intersex. When heterozygous, you have normal XX females, XY males, and they lack horns. But when homozygous, the XX animals develop as males. So we now know that this is work done in France, that this is definitely the causative gene. They use genome editing to specifically mutate FOXL2, and indeed you get this early gonadal sex reversal to give you animals with testis-like gonads, not ovary-like. In humans, heterozygous mutations in the gene give this syndrome, which I'm not even going to attempt to pronounce, BPES. It's autosomal dominant. No one's ever found a homozygous mutant, and the chances are pretty much zero that you would ever do so. You have eyelid malformations and sort of craniofacial problems in both males and females. But the females almost always show premature ovarian failure. So the gene has some role in ovaries. It certainly does in goats, and it probably does in humans. So we were interested in what happens in mice. And you also have the eyelid defects and things in mice. But if you study mice that are null mutant for this gene, so never seen FOXL2 protein ever, they have no phenotype in their ovaries. The ovaries develop perfectly okay. No phenotype is seen until about a week after birth, when the ovaries begin to become very messy, dysgenic, with sporadic upregulation of SOX9, which should only be expressed in the testis postnatally, and other male pathway genes. So we wanted to know what would happen if you deleted this gene in a less confusing period. Lots of things are happening this week, 2 weeks after birth. You have this period called mini puberty, all sorts of changes and things are happening. So what's happening if we just delete it from an adult female? Let's skip that. So we need a conditional mutation, an inducible Cree driver. This is the result. So we have, this is our control ovary, normal looking follicles, with a nice oocyte. If we delete this this was from an 8-week-old female, we delete the gene, and then we look 3 weeks later, just using normal histology techniques. You no longer have something that looks like an ovary. In cross-section, this looks a little bit like a testis. In fact, much like a testis, including structures that that look very much like seminiferous tubules, including cells that look like Sertoli cells, peritubular myeloid cells, and actually, I'm not going to give you the evidence, but there's Leydig-like cells in the interstitium as well. And these gonads are making levels of testosterone that are equivalent to their brothers. There's no germ cells because the oocytes don't like being surrounded by Sertoli cells, so they will die. But you've got gonadal sex reversal. So this is pretty interesting. It happens. So in fact, it happens very rapidly. So I'm not going to show you all the data, but if you look molecularly, as soon as you lose FOXL2 protein, you now see SOX9 being expressed. And the cells start transdifferentiating. So you have a transdifferentiation of granulosa cells to Sertoli cells, and then everything else follows on. We begin recently to, again, look a lot more molecular detail of what's going on, and clearly FOXL2 is, early on it's redundant. These other genes are probably helping to lead to ovary differentiation. So you don't need, if you just delete FOXL2, you still get ovaries. But after birth, there's something different happening. And so we think this is likely that FOXL2 regulates different genes at embryonic stages compared to postnatal stages through the interaction with different factors, probably bound to DNA at the same place and same time. So Roberta McGalley looked at this, is all recent work, and she used a very clever method which allows you to detect FOXL2 bound to its DNA target sequences, and also to identify the proteins it's interacting with when it's bound to DNA. And this has given a huge amount of data. I'm not going into it, but basically it says that the hypothesis is correct. FOXL2 interacts with different things at different times during development of the ovary. In the embryo, it's different compared with postnatally. It interacts with many more things postnatally, so it's becoming more important, we think. And then she looked, if you start looking at some of the factors it's interacting with, this is now revealing new candidates that are involved in disorders of sex development. So she found this gene, USB7. Again, not going to get any of the details, but just to illustrate the fact that this basic data is now giving us a lot of information which might help inform clinical cases. So if you delete USB in granulosa cells, it blocks primordial follicle activation, impairs ovarian development. We can't see sex reversal because the gonads disappear. They don't persist very long. It's such an important gene for ovary development. There are families living with USB7 mutations, and so clearly they have problems with fertility. Okay. And then this part of the talk, I just want to end with a little bit about, well, for some of these experiments, we end up using a lot of mice, particularly when you're having to get small amounts of tissue from embryos to do these molecular studies and genetic studies. We use quite a lot of mice. So of course we are interested in the 3 Rs and trying to reduce our animal usage. And so a couple of years ago, Nitsan in my lab started to develop methods for seeing whether we could derive gonadal cell types in culture from pluripotent stem cells. And she started this in my lab, and then we ended up collaborating with a group in Paris, led by Anu Bachambu, to see whether we could apply this methods in human as well. So it's very difficult to study gonadal development in human embryos because it's all occurring at a time when you can't see anything, it's inaccessible. It's very difficult. So if you want to understand a disorder of sex development due to a problem in the developing gonad, it's almost impossible. The only way we were doing it was by making mouse mutants and trying to see whether you got the same thing, but often it wasn't quite the same. So anyway, again, to rush through the story, we can begin with mouse embryonic stem cells or iPS cells, or human ES cells, and I'm going to show you a little bit of work with human iPS cells, where it's possible to begin with, if you like, control female iPS cells, control male iPS cells, and also iPS cells derived from XY female individual. And go through this process, which takes a couple of weeks, to differentiate the cell types that you'd normally find in the early gonad. You can then change the culture conditions, and you can follow various markers, and we can begin to find the markers that we expect to be present in the early gonad. We find that SOX9, which is our marker of cellular cells, is in the differentiating XY cell cultures, but not the control XX cell cultures. So this indicates that what we're getting is SOI-dependent expression of SOX9. It's the only real difference there. So this makes it a useful system, we believe, to look at disorders of sex development in humans. And just to prove this, we looked at this the iPS cells from this DSD patient, this XY female patient, and you could take them through to give gonadal cell types, but the cells from the DSD patient, which are the ones in the middle here, would never form tubule-like structures in culture, whereas from our control male they would, from the control female they wouldn't, obviously, but from the control male they would, from our DSD patient they wouldn't form the tubules. If you then use genome editing to correct the mutation in the gene, that we knew what the gene was, now they make tubules again. So we believe that this is a powerful system for studying DSDs in humans. Skip that. Lots of people I could thank for work over the years. I'm not going to go into all of these people now. I want to return to other things that involve in openness in this whole thing. So, as I mentioned, I was involved a bit in setting up the Concordat at its early stages. I mentioned talking about the sex-reverse mouse, Randy, a lot to media, and various other things that came up over the years that were relevant. And of course, Dolly was a very, very relevant appearance, because that was 1997. The paper was published February 1997, and it led to my first interview with Jeremy Paxman on the Tonight program. I survived. It's actually relatively easy. I was set up against someone else, and that other person couldn't make the arguments why you wouldn't clone animals very well, so it was relatively easy for me. So Jeremy was rapidly on my side, so that was pretty good. That led on to other things, and so in part it led me to being asked to help with changes to the Human Fertilisation and Embryology Act. So the changes were being made in 2000-2001 to permit the derivation of human embryonic stem cells and also therapeutic cloning, so like using the cloning techniques to derive embryonic stem cell lines, allowing that to be permitted in the UK. So I was corralled to help with that, partly because I was already talking to not the media, but also to some policymakers about these sorts of things. Anne McLaren probably also, who was sort of my mentor for a while on this, also pushed me into this. So that was all successful. So when it came to updating the HFA Act again, so this was, well, it started earlier than this, but basically 2 years of my life were devoted to this, 2006, 7 and 8, pretty much 3 years almost. It became necessary to update the Act again due to further advances in science, clinical practice, public opinion, etc., and also legal challenges due to ambiguous wording. So there were going to be many amendments planned to be made to the original Act, several of which were quite contentious. And just to say the punchline is that, again, it was passed by both houses of Parliament by a significant majority. But some of the contentious issues were things like pre-implantation genetic diagnosis and saviour siblings, research involving in vitro derived gametes, that's still contentious, and then human embryos carrying animal cells or DNA. So I was asked to join a working party by the Academy of Medical Sciences to look at the sorts of science that was being done and might be done where you have interspecies embryos, so where you might be mixing animal and human. This was rather focused on the sort of work that might be done using human embryos with some animal component. And particularly the things like hybrid embryos, where you wanted to do nuclear transfer using a cow egg and replace its nucleus with human nucleus. And that generated a lot of controversy and a lot of fuss. So anyway, we produced this report. It was initially entitled Interspecies Embryos, but actually the term that ended up being in the Act was Human Admixed Embryos. And we got in, it wasn't just about the hybrids, the nuclear transfer. We had 2 hybrids in there. We got transgenic human embryos, so where you've modified them in some way with animal DNA, and animal DNA could be interpreted very broadly. So it could be plant DNA or bacterial DNA. And actually, the genome editing that can be done now with human embryos, that was allowed because of these changes in 2008. Of course, we didn't know about genome editing back in 2008. So obviously none of these were going to be permitted embryos that could never be transferred back. So I was very much involved in the discussions, not just in Parliament, but with the media. And my view all along was to be very open and just say, this is what the science is about, this is what the science can tell us, this is what the science, how it might benefit us. No promises can be made, this is basic research, we don't know, but this is what it's about. So it got quite contentious, the debates got quite hot. If Fiona Fox was here, she would tell you all about this. So that Easter, March 2008, the Roman Catholic Church got very active trying to campaign against this. And basically the Cardinal Keith O'Brien condemned plans for the creation of hybrid human-animal embryos as monstrous. And we were likened to all sorts of individuals who were unpleasant. So I responded. Science progresses by... I'm going to read this out because I believe it. Science progresses by refuting falsehood. The bishops of the Roman Catholic Church at this time apparently want to promote falsehood. The proposals within the embryo bill that the bishops object to are to permit research which can be done with very sincere and honourable aims, which are to provide understanding of normal processes in life and to develop cures for the debilitating diseases and conditions that occur when these go wrong. Each of these aims informs the other. We are not seeking to make monsters, as the bishops can proclaim. If the research is successful in its aims, then it will help improve quality of life and give back dignity to those who suffer. Anyway, we won the debate. I wasn't alone, by the way. There were many others doing this, helping. Anyway, there's a report that followed up on that, which is perhaps a bit more relevant to understanding my own research. Having done the human embryos containing a little bit of animal stuff, what about the other way around? How about animals containing human cells? And so we wrote this report, Animals Containing Human Material, and that included, the first time I was involved in, a public dialogue. So this was not just simply doing public engagement, having consultations and things, opinion polls, but proper dialogue sessions with members of the public, which I think were really informative, and so I'm a great promoter of these. This was published in 2011. The day the report was published, a movie was released called Rise of the Planet of the Apes, which didn't help particularly, but I guess it helped the debate to some extent. And the recommendation took a while, but the recommendations of the report were eventually adopted into UK law, 2015. And then there are other situations where I've been involved in this sort of public dialogue, and it's very important. So, public consultation on methods to avoid mitochondrial disease for the HFEA. The Royal Society conducted a genetic technology program, which I was leading, and we did a public dialogue exercise to look at policy environment for the use of these techniques in plants and animals, as well as possibly in humans. So this is, again, genetic alterations. And then more recently, research involving early human embryos and research involving human stem cell-based embryo models. So these are very important ways of being able to, I know I've got to finish. Just go back to openness. So concordance and openness in our research is so important. I'm a great fan of open access publishing. I think it's really important. And for the scientists who are a bit resistant, because they have to pay a little bit more for it, they think, open access papers draw far more citations and from a broader readership than subscription-only journals. A supporter of open data, sharing of resources. Once published, I always give out everything, including genetically altered animals. So I think it's really important. Openness in clinical trials, of course, is also so important. So I'm going to, is that the last talk? Yes, that was the last slide. I'm going to stop there, just saying, be open with everything. Okay. So I probably should have been looking at what my clock a bit better.
No, it's perfectly fine. So I've got a microphone that's been through a lot, but I'm happy to give it to anyone that's got a question for Robin. And Hannah's got another one for there.
Yeah. Hello.
I'm Karen Gardner, formerly Glatzo, consultant to various organisations and worked with the IIT for a long time, and now work for the BBC. You and I worked together, especially with the IIT, and had many long discussions since the night about what stops, or what stopped people being open, and where the balance was between fear of their professional work being compromised and fear of animal extremism, which you and I both face directly.
Yeah.
And I've always said that the psychology around the animal extremism was far too successful because they were very good at endangering fear where there wasn't a lot of threat there.
Yeah.
And that seems to have changed now that people aren't so scared. What's your comment on that?
Well, you know, I was never particularly scared, and I never ever had any threat directly to me. Been open about this stuff for all these years. Not a single email. I'm going to invite them now, probably. I was told to check for bombs under my cars, all this stuff, and never did. No physical threat. We had these demonstrations outside the NIMR, but that wasn't personal to me. It was just they knew this was a place where stuff was going on. They didn't know what it was. And I think telling everyone what went on there would have been a solution to stop them doing it, because it was pretty much mostly mice and a few ferrets, and that was it, not much. And so the fear, I think, is often exaggerated, in my view. I know some people did receive threats, did receive nasty things. There were very nasty things happening to some individuals. And organizations were, and this still goes on, and I know that. But I think the openness helps, it can't hurt. And I think when people clam up and are not saying, you know, what they're doing and why they're doing it, it's a problem. But that's inviting trouble. I've actually had more complaints about the stuff I've talked about human embryo research. So I have had some nasty vitriolic emails and letters from that. But
any questions?
Gosh.
Yeah. Can I jump in on that?
Yeah, please.
So with the regulations the way that they are,
Yeah.
Does that preclude some of the science that you might want to do with hybrids, or that you can do? Are there some limitations there?
Regulations are, no, I mean, I think in terms of the the current Act needs to be updated again. The HFEA Act needs to be updated again, but that's not particularly to do with any animal research. And the animal regulations, I think, are in theory fine. There are some issues, and a lot of us, we were talking about these issues at an IET meeting the other day, President Advisory Group, where it's, you know, we miss seeing the inspectors, we miss seeing things like that. I think regulation could be smoother, where you're actually talking to the people regulating you in a much better way than that's occurring at the moment. There are some other issues which we find frustrating in the lab, actually getting the research done, because you know, we care a lot about our animals, but sometimes I think the steps we are forced to do are probably a little bit more stressful and harmful to the animals in the long run, because we have to have them under an operation for far longer than they really need to be. Just an example. But that's just sort of my personal experience and what people in my lab complain about. You know, it can be really hard sometimes to. So they are because this is too much for them. They don't have the time to spend doing some of the experiments. It's delegated to some really great people in our animal facility to do it. But again, what that means is that the PhD students and postdocs in the lab are a lot more hands off. And I think that's such a pity. So I want them to get back in doing stuff, but there's a barrier. There's, I think, some unnecessary barriers coming up to do this. But I may be only one person talking about this, but I care very much about our animals, but sometimes it gets a little bit silly. If there are any vets in the audience, they can attack me on that one.
Hi, Robin. It's Alessandro from the Royal Society of Biology.
Yeah.
What would be a potential candidate or like a sex-dependent disorder that you could treat in humans with genome editing in the future? Is there anything that you think may be coming up potentially?
Well, there's lots of cancers, which are, you know, some are being targeted already. There's, can I think on the spot about this? A disorder of anything in particular, or just?
Well,
there's metabolic disorders, there's, you know, all sorts of things that.
Do you think it would be quite challenging to kind of find a good targeting ways to do it in humans?
Okay, genome editing in humans, there are. Okay, I'm going to forget heritable genome editing for the moment. That has its lots of issues by itself. But gene therapy, the biggest issue is delivery of the genome editing components. So if you can take cells out of the body, manipulate them ex vivo, for example, bone marrow stem cells, which is done for beta thalassemia and sickle cell disease, for example. And then put the cells back through bone marrow transplant. That can work, right? It comes about with quite a lot of costs, including because you have to get the bone marrow graft to go in properly and take over, which means using nasty drugs to eliminate the resident bone marrow stem cells. And that almost inevitably leads to infertility of the individual and stuff. So really negative consequences. But the disease can be cured. And we've seen that now for sickle cell disease and beta thalassemia. The issues there are more to do with cost, equity of access. They're very expensive treatments done that way. If you could simply introduce them into the body so that the genome-editing components in a viral vector or a lipid nanoparticle, whatever, find the right cells to target efficiently enough, then that would be much less expensive, and it will allow you to hopefully target a greater range of diseases. You can get things into the liver quite easily, and that's proving to be quite successful as well. So there's quite a few metabolic disorders and things which you can now treat that way. But getting into muscle, getting into skin, getting into tissues that are difficult to access, the brain, really hard. So we need people working on delivery methods.
Well, thank you, Robin. One more round of applause for
Okay.